Plasmid Editor provides a dialog for facilitated manual PCR primer design. Plasmid Editor also offers a fully automated PCR primer design. You can find this tool in Wizard's menu. Clicking PCR Primers opens this dialog where you just need to fill in the required parameters and then click Design to let the program scan the sequence for all the possible primer locations. You can either select one amplification target region or two regions. If you provide just one region, the program will scan the sequence to find all the possible primers and products within the required length range. If you provide two target regions, the program will be forced to search for forward primers in the first region and reverse primers in the second region. I will demonstrate both cases. I will start with a trivial example of detection of EGFP gene DNA with PCR. I will first select the EGFP sequence by clicking on the feature label and then update the target region by clicking Update from Selection. Then I will click Calculate Limits to see what is the range of all possible PCR product lengths, and I will adjust this range to 300 to 500. Next, I will check the physical parameters of primers. I will slightly narrow down the tolerated range of annealing temperature. I will leave the other basic parameters at their default values, and I will also check the additional parameters. These values are the parameters of filters, the purpose of which is to prevent formation of undesired secondary structures or amplifying undesired targets. The primer-dimer stability score is a negative of predicted delta G value, and the default value of 5 is pretty stringent. However, if you have many suggestions in the output of the program, you can try to set even stricter value here. On the other hand, if you need to place primers in the sequence that contains a lot of self-complementary regions, you may need to increase this value. It's generally not recommended to have more than 3 successive G bases in the primer because it can lead to the formation of G quadruplexes, and also the primer synthesis yields can be lower. Long GC-rich stretches at the 3' terminus of a primer can lead to higher propensity to amplify off targets or to form stable dimers, so it's highly recommended to keep this filter at low stringent values. By default, the primer pairs are generated from a pool of candidate forward and reverse primers by random picking. That leads to a variety of primer pairs covering the entire target range and composed of different forward and reverse primers in the suggested list. Without this random picking, the program will output an ordered list of primer pairs, where, however, typically we'll see all the possible combinations of one forward primer and many different reverse primers, which is probably something you are not interested in. It may be useful in situations where you have very short amplification region and the program will output all the possible primer pairs. So when I'm done with the settings, I will click Design. The program will output the specified number of suggestions. With longer sequences, this is just a small sample of all the possible combinations of primers. For shorter target sequences or sequences with unfavorable properties in terms of GC content, sequence repeat, and so on, the number of suggestions may be lower than the limit. You can sort the table by product length or product location. These magenta numbers are line numbers that are not associated with the primer pairs. However, if you click one of these line numbers, the particular primer pair will be annotated in the sequence. This is the way the results are saved actually. So you have to pick a primer pair or more and click the magenta line number to annotate the primer pair in the sequence. You can then modify the names and other properties of primer annotations in this dialog. You can also see the predicted PCR product if you select the primer pair using click and control key plus click and pressing this button. You can also send the primer pair to Primer Blast to check for possible off targets if needed. Here you just need to select the right database and the correct organism and click Get Primers. I let the organism set to Homo sapiens, so this would make sense if I would express EGFP in human cells and wanted to detect it specifically on the background of human transcripts or genome depending on application. And here in this output of the primer blast are the possible off targets. However, in each of these results there are critical mismatches near 3' end, so these targets would be probably not amplified by this primer pair. Only in case there would be a long perfect match at the 3' end and only some mismatches close to the 5' end, only in such a case the off target could be amplified by the primer pair. In the last part of this video tutorial, I will demonstrate a case with two target regions. For simplicity, I will just select near the beginning of the EGFP cDNA and then a second region near the end of the EGFP cDNA. I updated the positions of the regions, and next I will check the length range of the possible PCR product, and I will leave these values as they are. Then I will just go on and click the design button to update the results. Let's say I have a preference for the longest possible product, so I will sort the table by product length, and it seems that the range of the length of the PCR products is a bit narrower than the theoretical range, which might be due to unfavorable composition of some regions in the DNA sequence. Also note that there are only 11 suggested primer pairs. I will just pick one of the primer pairs and click the line number to annotate it in the sequence. The reason for the low number of suggestions can be given by the GC content of the sequence. If I select the target sequence and check its GC content, I can see that the sequence is actually GC rich. This may cause that majority of primers within this length range have too high annealing temperature to pass the filter. So I can either increase the temperature limit to get more suggestions, or I could decrease the minimum length of the primers. Also in this case, I get many more suggestions.